Nature. Admired, observed and also shaped by people throughout history. A complex and dense network of interacting living systems investigated by science. A new field called synthetic biology is on the doorsteps to radically change our views about what life is, how it works and how it may confront us with living systems, products and applications that nature never could have created. Our film wants to take a brief look at the people that drive this field in Europe, who they are, what they do and why they're doing it.
My name is Markus Schmidt. I have a background in biology, in electronic engineering and also in the social sciences. So my role is to look into the wider societal implications of synthetic biology and in particular the one project that we are participating. We look into potential safety problems, for example, or ethical issues. We try to also make sure that the scientists that are working in the lab that are mainly interested in technical or scientific problems, that they also take into account that later on, when these scientific findings will be transferred into some products for the real world, they will also be safe and very good for society. The big driving forces of synthetic biology are some basic technologies or basic tools. One of them, gene sequencing. That means that they can read the information that is stored in the DNA. And the next thing is that you don't only read it, but you also write it. Make DNA de novo, new ones. No? So right now, there are the tools to make this DNA, to write information, but nobody knows what to write. So for example, you can compare this to the time of Gutenberg. So he invented the printing press. So he took, for example, the Bible and made lots of copies of it. But Gutenberg himself, he did not invent the Bible, he didn't write it. And Gutenberg also was not a poet. He was not Shakespeare, he was not Goethe. So what we don't have right now, the Goethes and Shakespeares of biology.
In May 2013, life scientists from all over Europe meet in Elmau Castle, Bavaria. They were participating in the EuroSynbio program, 5 different projects that were funded by the European Science Foundation. The aim is to support the development of synthetic biology within Europe. The meeting in Elmau gives them the opportunity to exchange the outcomes of 3 years of scientific work.
So science lives from free exchange of ideas, as free as possible. People read and they know a lot about their home turf, but in order to get new ideas, it's important to come and hear what others do, even if it's in a remote field or so. Very much of this is just initiating new ideas, different angles that one didn't explore before, stuff like that.
The meeting is first, it's a scientific exchange which we do here. We are looking for how to continue the research which was funded for 3 years, which was seen as a seed, as an initiation of to establish synthetic biology in Europe.
Synthetic biology approaches life with the eyes of an engineer. Organisms are seen as living systems that consist of functional parts, parts that ideally are reliable, clearly defined, and that stick to certain standards and rules. The cell is therefore seen as a kind of factory hall for chemical machines that digest and assemble matter, and that are able to produce a desired product.
Well, you can consider a cell as being a a huge factory. So we have proteins doing the communication, other ones are transport, other ones transport trash, other ones generate energy, which is a currency for paying work in the cell. And so what we do is we take these machines of the cell out of the cell and we use them in a synthetic environment so that they can do the work we want them to do.
Initially, Daniel Müller was interested in proteins, the molecular machines that drive the cell.
And at the beginning, I never thought about engineering some proteins. I investigated them and I just watched them and tried to understand how they function. And then slowly, I became aware that you can modify and manipulate your proteins. You can direct the proteins. You can guide them to do what you want, or you can use them to do what you want. You know, if you investigate something, you try to understand it, and as soon as you understand it, it opens, or you think you understand it, it opens the eye for an application, and then you do something new.
Müller wants to create nanocells, thousands of tiny fat bubbles that can host his protein machines. The aim is to establish a reliable and predictable biofactory on a nanoscale.
You know, the idea to build a nanocell was the follow-up process of an EU project where we built motors, molecular motors. And the next was then, why not bringing these motors or the machines together into a compartment, and which compartment could we use? And that was the nanocell at that time.
The molecular machines already exist. Nature provides proteins for an almost unlimited number of different tasks. They just have to be found.
What we have to do, we just have to go to literature. It's reported which kind of different proteins are there. And then we look to this enormous catalog of proteins, this portfolio the nature has generated for us or provided for us. And then we pick out certain proteins, which are our machines, which are the molecular machines of our nanocells, and we put them together so they can work in synchrony in the nanocell.
The nanocells themselves are assembled by an automated device that was also developed by Professor Müller and his team.
This machine here, it folds these molecular machines into the nanocell. So what it does, it has the nanocell membrane and folds individual machines into it and equips the nanocell with function. At the moment, it's much more fundamental research. Is it principally possible to build up a molecular factory? What do we have to learn to do it? Which tools do we still have to develop to build molecular factories?
There are many ways in which nanocells could be used in future. The desire to do so, to switch from fundamental science to industrial application, is on both sides.
As soon as we have the first molecular factory, then they will possibly try to take some of this knowledge and then use it by themselves, which would be fantastic.
The University of Groningen in the Netherlands, a brand new lab for brand new antibiotics. Professor Oscar Kuipers develops them using the methods of synthetic biology.
When I was studying biology, I was interested in plants and plant physiology and so on, but suddenly I took a course in microbiology, so I got very excited that you can work so quickly with bacteria that every day you can do experiments and the next day you already see the results. So from then on I was really hooked on microbiology at the molecular level, so to really understand what's happening in bacteria with their proteins, with their DNA, and how things function in bacteria. These labs here on the left are mainly the antibiotic researchers, so there are 7 people working on antibiotics, and in the rest of the group people are working on other subjects like single cell analysis and heterogeneity in cultures.
Are you working in the lab as well?
No, the last thing I did in the lab was 3 years ago when one of the technicians trained me how to do a microarray. I just wanted to know how that was done, and before that I did 10 years nothing in the lab, I think.
So what are you doing?
I'm mainly managing the group, I think, and making sure that we get enough grants for new research programs. And of course I'm talking as much as possible with the PhD students and with some of the postdocs about the progress in the research. So you have to be able to manage well, and also to be good for your group, I think, and but also to be a good scientist. Otherwise you don't have new ideas for new avenues. So I think you should combine all these skills and train yourself in the skills that you don't already have. In Europe, and especially in the Netherlands, people look very much at the utility aspect of research, and also that is true for getting funding. So it's my conviction that you cannot always first define the application and then define the research that should lead to that application. It also works the other way. Just try to work on the borders of science, on trying to get into new concepts, and then look how that could be applied.
The application Oscar Kuipers is working on is desperately needed, especially in hospitals. A growing number of bacteria become resistant to antibiotics. A natural outcome of evolution that cannot be stopped, but only be addressed by research on new substances.
Infections occur more frequently. So we have now to get prepared to what will happen in the next 20 years.
Although desperately needed by medics, industry refuses to do research on antibiotics, as it just does not pay off.
There's not really a good economic incentive to produce develop over 10 to 20 years a substance that at the end you use for 1 week or 2 weeks maximum, and then if you use it a lot, it will not work anymore. If you look at how many antibiotics have been brought into the market in the last 10 years, you will see that there are very few ones, and in the next years it will be even worse.
As it is unlikely to find new antibiotics in nature, Kuipers hopes to be able to create synthetic ones. His hopes lie on a group of well-known peptide structures called antibiotics.
Well, antibiotics is a class of antimicrobial peptides, so they are very well known in literature. We know more than 100 of these structures, which all have different kind of ring structures, and we take these ring structures and combine them in all kinds of different configurations, so that we can make millions of novel peptides. And then the idea is to screen these peptides and to find the best working and the best active and the most stable peptides against the most dangerous pathogens, or those pathogens that have a lot of resistant variants.
The efficiency of an antibiotic substance can directly be tested in a Petri dish. Antibiotic peptides build so-called flares, spots where other bacteria cannot grow anymore.
If we find really the nice targets and we characterize them and we can say, yes, this is a very good agent against that bacterium, I would say from a well-characterized peptide with perfect activity, it would still take at least a couple of years, perhaps even 3, 4, 5 years before it is fully tested and found to be safe, and then it can be put on the market.
People know Norway for being a rich country, at least in oil and gas. Trygve Brauteset from Trondheim has to deal with that.
Some people come to me and they say, well, in Norway it must be good to be a researcher because you are so rich. So funding must be extraordinary. No, it's not. It's rather poor. Many Norwegian researchers are a little bit more static. We stay in Norway, but there is more and more force towards international collaboration in projects. So we are getting more and more international in our research, and that is what we want to be. But still, we are a little bit outside the rest.
Although not a member of the European Union, Norway decided to join the European Science Fund. The SINMET project is investigating a special kind of bacteria.
In Norway we are very rich in natural resources, oil in particular, but also gas. And from gas you can make methanol. But in our case we use bacteria that can really eat methanol. Most microorganisms cannot do that.
Brauteset's bacteria could process abundant natural gas into useful compounds.
So the SINMET project deals with development of microbial cell factories. Let me illustrate this. So here is a microbial cell. A microbial cell can make a wide range of different products. These can be antibiotics, amino acids, feed supplements, and so on. In order to make this running, the bacteria needs a substrate, something to eat, so they can grow and divide. This is typically today sugars. So you feed them with sugar and then they produce your product. In the SINMET project, we are not so much taking care of the product. We are interested in the substrate, that is what the bacteria can eat. And today there is a huge pressure on using sugar for food instead of using it for biotech industry. And therefore we are aiming to create bacteria that can use methanol instead of sugar. So you can say that you replace sugar with methanol to have cell factories that can convert methanol into useful products. You can engineer completely new properties into microorganisms. You can even completely reprogram microorganisms by using synthetic biology. This is the molecular biology lab. So you have to build up a team with people with different complementary expertise. In order to have a project running in a good way, you have to have regular meetings to discuss what results have we reached so far, what do we do, do we have to change our plan. There are always things that doesn't work, so you have to readjust your plan constantly. We have a long tradition working with microorganisms to genetically modify microorganisms to make cell factories. So here is where most of the genetic engineering work is going on. Genetic engineering, that is to take the DNA out of the cells, to play around with the DNA, recombine the DNA to make new pathways and put them back into the bacteria. This is the fermentation laboratory. So this is where we cultivate the genetically modified bacteria that we make. So we do the genetic engineering and then we test them in a kind of small scale. And when we find some very interesting bacteria, we really want to test their performance and properties in the fermenters like here. Inside the fermenter you have what we call the growth medium. That is water, and then you add minerals and vitamins, things that the cells need in order to grow, and then of course the carbohydrate source, which we call the substrate or the raw material, which here is methanol.
So this is a kind of alcohol cocktail?
It's a kind of alcohol cocktail, yes, that the bacteria can grow.
Conserving information needs a code and a medium. Throughout history, mankind developed many different media and codes to do so. Nature only uses one code, the DNA, which was discovered at Cambridge University by Francis Crick and James Watson in 1953.
The revolution in biology, in molecular biology, which started with the discovery of DNA, is unprecedented also in the history of science, the speed with which this has developed. I think people have underestimated the complexity of the genome. There's many, many more layers of control and regulation involving all those parts of the genome which previously were thought to be, boring and non-coding and unimportant, sort of junk DNA, what it was called. I think the whole machinery is much more complex, much more sophisticated than had been anticipated.
60 years after the discovery of DNA, again in Cambridge, Phil Holliger and his team achieved the engineering of a biological storage system. From now on, genetical information can also be stored within XNA.
So if we think of the DNA molecule, the very simplest form as a ladder, I mean it's not a straight ladder but a ladder that is twisted. We are not changing the rungs of the ladder, so what you would step on, but what we're changing is the scaffold on which the rungs are mounted, so the backbone of the DNA. So that's the difference between DNA and XNA. It's not in the rungs, but it's in the scaffold. And this is important because what the rungs do is that's where the information is encoded. And the scaffold is really just a chemical framework that holds those letters in place. So that means we have a different medium to encode the same information as we have in DNA. And that's how we can read information from DNA into XNA and back into DNA.
And all of these mechanisms, they already work.
And they already work, that's why we're here.
It's not just a concept?
No, it's not just a concept. No, we have been able to do this. So at the moment it works at a purely chemical level, but in a way all the pieces are in place to maybe at a later stage, if you want, invade biology and set up a genetic enclave within the cell, which could be encoded within XNA and could communicate with the rest of the cell. We use actually very, very simple tools like centrifuges and pipettes, and really it's sort of iterations of very, very simple, actually very low-tech tools, but used in the right way, they can be very powerful.
Petra Schwiller is a physicist. She has a simple but essential question. How do cells divide? Here at the Max Planck Institute in Munich, Bavaria, she may find the answer.
So I'm here since last summer, but the institute is already a very established one. Well, it has a long and interesting history. It's a very large institute, one of the largest of the Max Planck Society. Hi, so. Yeah. So this is my workbench, which is unfortunately not the most organized one at the moment, but the organization is all in my head, of course.
This is where top-of-the-edge science happens.
This is where top-of-the-edge science is conceived, so to speak. We are trying to understand fundamental biological processes with the eye of a physicist. which is completely a different eye as the eye of a biologist. So if you want to describe a biological system, if you are a biologist, you want to understand it basically in detail. You want to understand every single part of it, and you want to describe as close as possible what you find. The physicist has a different attitude. The physicist wants to understand the principle. They're not so interested in the details. They want to understand what is the driving force, what's the principle, how could I basically simplify what I find as much as possible, but still having the same phenomenon. And this is what we want to do with cell division. We want to, or cells in general, we want to understand the essence of it.
As cell division happens on a molecular level, it is necessary to have the tools to be able to watch single molecules. A special method called laser microscopy can do the job.
This is our laser lab. We are basically developing imaging and spectroscopy for single molecule investigation in the cell. The fluorescence technology allows you to label any molecule that you want to see. I really want to know how complicated is this mechanism. This is really what is my genuine interest.
The so-called Min proteins are assumed to be a key player in the division process. They can be observed moving back and forward within the cell, defining the point where division happens.
You can actually see that they oscillate back and forth on a very, very small pace. We can also see how the divisome assembles at the mid-cell, which is basically culminating in a ring, a contractile ring, as we believe. And the contraction of this ring is then supposed to split the two daughter cells apart.
For easier observation, Petra Schwiller and her team were able to get the Min proteins working outside a natural cell. A first step towards a bigger goal: the synthetic cell.
So this is a system which is mimicking events in cell division of bacteria. So bacteria divide, E. coli bacteria divide by oscillating, having a specific protein machinery oscillating back and forth between the two poles, like this like cavity, which is not a bacteria but some mimicry of a bacteria. And these proteins basically, by oscillating back and forth, they define the axis of division, which is the mid-cell, so to speak. And another group of proteins, not the oscillating ones, but other ones, they will be able to assemble in the middle, because this is the space where these oscillating proteins are least of the time. And then there's a ring assembling in the middle, and that ring somehow miraculously contracts. And this is what we want to reconstitute, this contraction and the division of a compartment. If you think hard enough and if you do research, you will at some point find how from the inanimate, the animate will basically emerge. And I don't really have a problem with that. I don't feel like being God or playing God. For me, I have to say that God is more than just the creator of life.
It's quite difficult to foresee the future, and what we're doing right now is we try to make sure that it's transparent, it's reflective, and it's taking into account wider societal issues, so that when we see something ahead of us, we are able to stop and to reconsider and change path and go a different way.
We have come a long way. A lot of things can already be done, and we have usually rather strict regulations, what should be done, what can be done, what should not be done.
It is a matter of what is being made and what is being achieved. I think that when people see a benefit from a new technology, the acceptance level is much higher.
Well, if you investigate, you never know exactly what you're going to find. So if we stop analysis of genomes or interpreting analysis of genomes and doing experiments on that, we will also miss a lot of knowledge which could be very beneficial for ourselves.
Humans have been creating, right? They have been creating ingenious artwork. They have been creating symphonies. They have been creating machines. I think creation is not reserved for God.